Hello, everyone. My name is Korbin Biswas, and today I'm going to explain a scientific lab technique called Western blotting and how it is used to make modern impacts on society. First, I'm going to explain what Western blotting is, then how it is used, and finally provide a real-world example. So to begin, what is Western blotting? Western blotting is a method that scientists use to detect specific proteins in a given sample. Proteins are important molecules because they are the basic building blocks of life and perform many functions, like controlling muscle contractions, heartbeats, and signaling within the body. Western blotting allows scientists to measure how much of a certain protein is present and how cells are functioning. Now, here's a brief explanation of how it works. First, proteins are sorted by size in a gel using electricity. Next, proteins are placed into a thin sheet called a membrane, so they're easier to study. After, the membrane is covered with a protein solution to prevent any unwanted sticking. Fourth, special molecules called antibodies attach only to the target protein the scientists are trying to reach. Finally, a signal, often in light or color, displays the protein as a band. The darker the band, the more protein is present. I also include an image where you can see the difference in the bands. The dark bands contain more proteins. To see how this technique is used in real life, I wanted to point your attention to the study titled X chromosome linked microRNAs regulate sex differences in cardiac physiology. In this study, scientists were interested in a protein called SRL, which helps control calcium levels in heart cells. Calcium is important in heart cells as it helps the heart contract and relax properly. Using Western blotting, they're able to compare how much of this protein was present in male versus female mice. They're able to see that male hearts demonstrated more SRL protein than female hearts. To learn more, the researchers wanted to understand why females had less SRL protein than males. They discovered that females had more of a molecule called MIR871, which reduced the amount of SRL protein. This difference affects how the heart resets between beats, what we call repolarization. It also explains why female hearts show different levels of heart rhythm issues. Without Western blotting, these scientists would not be able to clearly see this difference in protein levels between the sexes. Finally, I wanted to explain why you should care. Heart issues like this have impact many patients across the country. Research like this helps potential parents learn more about the risks of heart disease associated with having a particular sex. Additionally, it helps parents and other patients learn more about potential treatments for life-threatening diseases. These discoveries would not have been able to happen without techniques like Western blotting. Thank you for listening to my presentation, and I hope that you leave more informed.